Hello friends, in our new video we will study meiosis. Meiosis aims at maintaining a fixed number of chromosomes for various species during succession of generation where gametes, eggs, sperms in animals are created and spores in plants. As we see, the gametes are all formed by meiosis. Meiosis is two successive divisions, means as we study in mitosis, interphase, and then after interphase, mitosis takes place, and after a new interphase, a new cell division. But in meiosis, one interphase, after interphase, two divisions takes place and divisions stop completely. No any gametes can divide by meiosis or mitosis. Meiosis is two successive divisions of cell. During this division, the number of chromosomes is reduced to the half number of somatic chromosomes. Because there are two divisions, genetic material one times duplicate in interphase, but two times divide, so result says they have half number of chromosomes according to somatic cells. When two nuclei or gametes are fused by fertilization to form embryonic cell known as zygote, again total number of chromosomes obtained. That's why they call meiosis maintain a fixed number of chromosomes. How? By one forming half number of chromosome gametes, and this gamete fusion or by fertilization for new organism with total number of chromosomes. We said meiosis consists of two divisions. Meiosis is done through two nuclear division. The first separate homologous chromosomes. Homologous chromosomes, as you remember, we said there are chromosomes like this before cell division, but they only appear during cell division. But these chromosomes, they are duplicate to form what? Two chromatids. These two chromatids of same chromosome, we call them sister chromatids. And also that chromosome can copy itself, which made up of two chromatids. Also they are sister chromatids. But these two chromosomes, they have similar functions. How similar functions? A part of that chromosome, if contain information about eye color, same part of that chromosome also about eye color. Maybe blue eye or black eye, but both of them for eye color. Another part of chromosome also similar, contain genetic information about hair color, or about part of chromosome about, for example, ears, and another part of body, and so on. These two chromosomes, which have similar functions, we call them homologous chromosomes. This homologous chromosome, one of them received from male and one of them received from female. And in previous page we said meiosis provide fixed number of chromosomes. So half from father and half from mother, so new organism totally has what? Total number of chromosomes, which we call it zygote. But during meiosis 1, these two chromosomes, they separate one from another, and each cell receives one of them. So said in first one, homologous chromosomes separate. But in meiosis 2, which also consists of 4 phases, which have similar name we study in mitosis, that time chromatids are separate. That's what these two chromatids separate one from another, and also there's two separate one from another, and each cell receives one of these pieces. A major difference between meiosis 1 and meiosis 2, in first one homologous chromosomes separate, but in second chromatids are separate one from another. The first meiosis division has similar phase like prophase, metaphase, anaphase, telophase. For meiosis 1, we use number 1 like prophase 1, metaphase 1. For second meiosis, prophase 2, metaphase 2. We remember from mitosis that the duration of phase is depend on how much changes happen in that phase. So in prophase 1 of meiosis, because so many changes happen, the duration is longer according to prophase in mitosis and also prophase 2. So depending on this so much changes, the prophase 1 is divided into 5 subphases: leptotin, zygotin, pakitin, diplotin, and diakinesis. What first we need to know, these names in order, and after we share changes between these subphases. In the first phase, leptotin, like an early prophase, chromosomes at this round are single. Single means, you remember we said there are homologous chromosomes. Homologous chromosomes, single mean they are far away from one another, these chromosomes, so that they are single. They don't make pair yet. And the long filaments, like threads, they look like necklace. We know in nucleus, before cell division, there is genetic material in form of long filamentous shape in that form. And filament-like bits. So this genetic material is hard to separate and share between two cells. So gradually, step by step, the genetic material condense, coil around itself, shorten, thicken to form chromosomes. But in leptotin stage, said they look like necklace. That's what the chromatin network extend, coil around a protein, extend again, coil, extend, coil, extend, coil, extend, coil, extend. So in that way, it looked like a necklace. In which round? Leptotin round. But what important? Chromosomes are single. They didn't make pair yet. Genetic material is doublet in each chromosome. Doublet, we know genetic material doublet in interphase. But in interphase, it's not so clear to know if there are double or two pieces of genetic material which are similar. But as chromosome formation started, we can identify there are two parts of same chromosomes, as we call it, chromatids. In next phase, zygotin, homologous chromosomes align and double. So that genetic material align and double. Coiling, as we said, like next phase, it increases. Coiling increase, genetic material coil around itself, form a shape like this. Homologous chromosomes align and double at this round, then they bend to each other at that point. This process is called synapsis. Synapsis, as we said, they are single, far one from another. By step, they come closer one to another to make a double, to make a pair. Name of this process, we call it synapsis. But that structure, which is made up of two chromosomes, we call it bivalent. So the doublet chromosomes are in zygotin round of prophase 1, we call them bivalent. This process is distinctive in meiosis because it does not occur in mitosis. There is no pairing of homologous chromosomes in mitosis. In the next phase, packeting at this round, chromosomes condense, shortening and thickening increases. Each chromosome double into 2 clear chromatids. Because shortening and thickening increases, at this round we can see maybe a shape like this, chromosome made up of 2 clear chromatids, and another chromosome also consists of 2 clear chromatids, but bend to one another in some points. Each 2 chromatids of the same chromosome are called sister chromatids. These 2 red-colored pieces, they are sister, and these 2 blue-colored chromatids, they are also sister chromatids. At this round, each pair of homologous chromosomes form a band of 4 chromatids, and we call it tetrad. So this band of 4 chromatids, we call it tetrad. How in zygote stage we call them bivalent, but as shortening, thickening increases, they become clear to see, and we call it tetrad. So band of 4 chromatids, or which consist of a pair of homologous chromosomes in packeting stage of prophase 1, known as tetrad. For example, human cell at this round has 23 tetrads times 4 equals to 92 chromatids in total. In addition, at this round, there is exchange of gene locations between identical chromosomes. The identical chromosome means homologous chromosomes. They are not in genetic information identical, but as you remember, we said they have similar functions. Like what? Part of that chromosome for eye color, part of that chromosome for eye color, also part of that chromosome for eye color. So these 2 similar parts, which both of them for eye color, but one of them may be blue eye, one of them black eye. These pieces are exchanged between 2 non-sisters, means one red and one blue. Exchange of this dislocation is called crossing over. And crossing over is a process which helps to form different cells in meiosis. So question asks, why different cells formed or non-identical cells formed by meiosis? One of these reasons is crossing over. In diploid stage, at this round, the homologous chromosomes repulse and part. The non-sister chromatids remain attached by 1 or more points. At this connection points are called chiasmata. You remember in previous one we draw chromosomes like this, and we said at some points these chromosomes, they connected one to another. The connection point between non-sister chromatids, we call them what? Chiasmata. Number and location of chiasmata differ from chromosome to another, and in some chromosome maybe only 2, maybe 4, maybe 5 more connection point between non-sister chromatids are known as chiasmata, differ from one cell to another, from one cell to another. Non-sister chromatids are exchanged at each chiasmata point, which we call it crossing over. In diakinesis stage, this is the last round of prophase, at which chromosomes of chromatid homologous chromosomes shorten and thicken more. The nucleus and the nuclear membrane gradually fade. They also happen in mitosis. Location of chiasmata move to the far end of the chromosome, thus chiasmata decreases. Chiasmata decrease means these 2 chromosomes connected to one or more part one another. By step, they separate one from another, but they remain to stay closer to one another again, but only connection point between them separate. But as they separate one from another, we can identify some parts are exchanged between these chromosomes, which we call it as crossing over. So after these pieces are exchanged between them, the same pieces of chromosome also, because they are different than before, we no more call them sister chromatids. Because sister chromatid means genetically identical chromatids. But as they exchange some pieces, they are not identical of their nearby chromatids, so they are no more sister chromatids, but just we call them chromatids. So chiasmatic decrease in 4-5 point days connect one another, but as time passed, 5 and then 4, then 1 more separation, 3 and 2 and 1, and at the end they totally separate one from another, homologous chromosomes. Metaphase 1, at this phase, homologous chromosomes align at the equatorial line in the form of binary chromosomal groups. If we draw a cell in that form, we draw chromosomes 1 and 2, but with some exchanged pieces. They align on the equatorial line of plant, connected to the spindle fibers by their centromere, centrosomes at the poles, but binary chromosomal group and 2 chromosomes together. Maybe if there are 1 more pair of chromosomes at that site, also 1 more tetrad we can draw, but also with what? Some exchanged points between them, which takes place in Pakiti's stage of prophase 1 by crossing over prophase to form cells different one from another. Centrosomes start to appear and the spindle fibers are attached to centrosomes or centromeres. Anaphase 1, at this phase, homologous chromosomes separate. As you remember, we said in meiosis 1, homologous chromosome separate. So if we show on that picture and we draw a cell again, centrosomes, spindle fibers, homologous chromosome separation means 1 chromosome totally to that side, but it's homolog to the next side, and 1 other chromosome, to one side, and the next chromosome, it's homolog to the other side, but they have some exchanged parts. We need to show them for what? For crossing over. So, main difference between anaphase 1 and anaphase of mitosis or anaphase 2 is separation of homologous chromosomes. And move in opposite direction toward to the cell poles, chromatids of each chromosome remain to each other by their centromeres. In telophase 1, new chromosomes gather at the poles. Spindle fibers often disappear. They may not disappear too. The nucleolus and nuclear membrane which surround the chromosome start forming, but before formation is complete, next division starts. This group is haploid. Haploid by 2n and n. If it is n, we call it haploid. Haploid means there is only one chromosome. With what? From the pair of homologous chromosomes. Or if they make a pair, homologous chromosomes are together in the same cell, we call it diploid. Means what? If there is only one of the homologous chromosomes haploid by itself, that also haploid by itself, but both of them, these two homologous chromosomes together, we call it diploid. But because in anaphase homologous chromosomes separate, now the groups are, or chromosomes are haploid, because they contain half total number of chromosomes. So being haploid or diploid, not about numbers like 23 or 60, 46, but depend on what? Depend on homologous chromosomes. If homologous chromosomes, like a pair found in a cell, this cell is diploid. But if they separated one from another, cell contain only one of them, it's haploid. Contains half of total number of chromosomes. Nuclear division is followed by cytokinesis. Cytokinesis, as we study in meiosis, in plants by formation of cell plate, and also in animals, but furrowing from outward to inward, pinching of cell and cell divide into two. They are ready for the second meiosis. As we see, as telophase 1 ended after cytokinesis, the next division starts without interphase. So we call meiosis consists of two successive divisions. Second meiosis, prophase 2. When we study on second meiosis, as we start synthesis, we're going to compare it with the previous one, with the meiosis 1. So the number of chromosomes in each nucleus prophase 2 is half of the total number of chromosomes, but while in prophase 1, total number of chromosomes. Reason? Because in anaphase 1, homologous chromosome separate. It differs from mitosis, in which the nucleus has total number of chromosomes, and also for prophase 1. At this phase, the chromatids are distant and might differ in structure. Differ in structure because of crossing over which happen in prophase 1 of meiosis. Now, these two chromosomes, they are in different cells after cytokinesis, but they are no more sister because of what? Because crossing over which happened in prophase 1 of meiosis. At this phase, the chromatids are distant, may differ in structure because crossing over outpacketing round of prophase 1. Metaphase 2, at this phase, the chromosomes are located at the equatorial plate of cell. It is attached by spindle fibers through their centromere. So, if there are spindle fibers and the spindle fibers, but at the spindle fibers, only what? One chromosome. On that side also there is only one chromosome with exchanged parts. Each chromosome is made up of what? Two chromatids. While in metaphase 1 of mitosis make binary chromosomal groups. That's what? These two chromosomes, homologous chromosomes, they were together in metaphase 1, but in metaphase 2 they separated and they are in different cells. This phase is differs from anaphase 1 in that chromosome in metaphase 1 are 4 chromatids, while in that phase consists of only 2 chromatids. A major difference between metaphase 1 and metaphase 2. Anaphase 2, at this phase, chromatids of each chromosome split. You can see we said chromatids of each chromosome, but we don't use the term of sister chromatids because they are no more sister. They are different in structure because of crossing over. When their centromeres separate, each chromatid represents a chromosomes. And after they apart one from another, we draw in this shape. For example, one chromosome, chromatid, another chromatid, and also chromatid and chromatid, but with one of them has different genes. Also, that one is different. When their centromeres separate, each chromatid represents a chromosome. And after chromatids separated one from another, each of them, we call it chromatid. Chromosome, independent chromosome, independent chromosome, and independent chromosome. Telophase 2, at this phase, chromosomes gather at the poles. They get longer and thinner. Chromatin matter appear as fine threads, then the nuclear membrane and nucleoli emerge, and which in telophase 1, nuclear membrane and nucleoli emerge. We said they start to appear, start to form, but before formation, complete next division. Start, but in telophase 2, nuclear membrane form, nucleoli form, spindle fibers disappear totally, which will not disappear in telophase 1. In some plant cells, the cell plate is formed through the spindle and become middle lamella. We remember middle lamella from one of 3 components of cell wall. As for animal cells, cytoplasmic membrane is generated between the 2 new nuclei and separate them, similar like in mitosis. When the first and second meiosis division ends, the total outcome is 4 cells with haploid chromosomal group. And we draw the cells F1, 2, 3, and 4. Each of them receive one of these chromosomes, but they all different one from another. Because of what? Because of crossing over which happen in prophase 1. So, question can be asked, why 4 cells formed in meiosis but 2 only in meiosis? 4 cells in meiosis, because meiosis consists of 2 successive divisions. Why haploid chromosomal group cells are formed? Because homologous chromosomes separate in meiosis. And also why non-identical cell formed? Because there are 4 cells, how they are different one from another, also they different from parent cell. That's because crossing over happen, and also homologous chromosomes separate. Meiosis occurs in testes. They are gonads, means reproductive organs of male in which reproductive cells are formed. Ovary, gonad, which produce gametes. Female gametes are formed in animals. It occurs in plant cell, also eggs, and immature male reproductive cell pollen formation. Although there are some differences which will be, discussed later in reproduction part. Mitosis and meiosis comparison. Mitosis has one division, that's why only 2 cells formed in mitosis, but meiosis 2 successive divisions, that's why form 4 cells. 2 identical cells formed, as we study in mitosis, new formed cells in amount and type of genetic material, they are same. But in meiosis, 4 non-identical cells formed. We said why they are non-identical, because crossing over happen and homologous chromosomes separate. Genetically identical cell formed, why? Because chromosomes not separate, but chromatids, sister chromatids separate in mitosis, but genetically different cell formed in meiosis, because in meiosis homologous chromosomes separate and crossing over occur. Number of chromosomes in both new cells similar to mother cell. Why? Because genetic material duplicate in interphase, and after there's only 1 division. But in meiosis, number of chromosomes of the new cells are half of mother cell, because there's 1 interphase, 1 duplication, but 2 divisions. Somatic cell divide in mitosis, like in skin, epithelial cell divide to form epithelial cell, or neurons divide to form another neuron. But in meiosis, reproductive cell form, not divide, they form by meiosis. Somatic cell divide to form somatic in mitosis, but somatic cell divide to form reproductive cell in meiosis. Division occurs during the cell life continuously, yani the day fertilization happens, zygote pass through mitosis to form 2 cells, 4 cells as organism grow, and they need body part to repair, mitosis occur. Division of meiosis occur after sexual maturity, where organism gain able to produce reproductive cells. Mitosis division used for growth, because we know 1 cell come from another cell by means of cell division. As number of cell increase by division, organism grow. Repair cells, as broken bones, scars in skin, they all repair it by forming new cells by mitosis. And asexual reproduction, like in amoeba, which consists of only 1 cell, pass through mitosis, but form 2 amoeba, and it's kind of what? Kind of reproduction, because an organism which similar to mother cell is formed, which call it reproduction, but because there is no reproductive cell formation, nor to fertilization, it is asexually reproduction. Meiosis division is used in sexual reproduction and having new members of that species, because produce gametes, and gametes after by fertilization form a new organism. Meiosis needed for sexually reproduction.